Afternoon, everybody. Our speaker today is Sam Hess, who obtained his BS summa cum laude from Yale University and then studied physics at Cornell University, where he received his PhD in 2002. He was also a postdoctoral fellow, fellow at the NIH. He's now associate professor in the Department of Physics and Astronomy at the University of Maine. His research interests include nanoscale membrane organization, influenza virus infection, and photophysics of fluorescent protein and quantum dots and super-resolution microscopy. In 2006, Dr. Hess published the invention of Fluorescence Photoactivation Localization Microscopy, FPALM, which circumvents the diffraction limit in which light microscopy can be used to image subcellular structure with nanometer resolution, 20 to 30 nanometer. And this is the topic of his talk today.
Thank you very much. Can everyone hear me okay? I would also encourage anyone to ask questions during the talk. If anything could be made clearer, I'd be grateful to take questions. And I'm also very grateful to have a chance to present our work. I think it's an exciting research field and has a lot of potential for forwarding science, especially biomedical. So as Alex mentioned, our technique is able to break the diffraction limit, and so I'll describe first what I mean and how the technique works. So it's called FPALM, or Fluorescence Photoactivation Localization Microscopy. I'll discuss some biological applications that we're working on and that are quite possible. And then there are some additional versions of the technique that, including three-dimensional imaging, that have been recently developed and can also help in imaging biological systems. And in principle, this can be applied to any kind of a sample. It doesn't have to be a biological thing. So this started with the classic problem that diffraction limits resolution in light microscopy. And since biological structures of interest are on the nanoscale, we would really like to be able to get past this Rayleigh limit of about 200 to 250 nanometers in light microscopy. Of course, you can use electron microscopy, but it's hard to do live cell imaging with such methods. And so the concept is that we would like to keep, if things are closer together than are zero within a sample, then even though the object may be point-like, its image is finite in size. And so if two objects are closer than are not together, they're very difficult to separate as distinct. And so the structure in the sample is blurred on length scales less than R naught. And this is getting in the way of lots of interesting studies. So also to point out that this problem has been around for some time. This is Ernst Abbe's memorial, where the diffraction limit is actually carved in stone. So this is something that's been around since the 1800s, and we're trying to break this rule that's carved in stone. So the beginning of getting around this limit is the concept of localization, and that is to say that you could have objects that are separable from one another. That's resolution. But if an object is by itself, you can image it and determine its location quite precisely. And so that's localization, finding the position of an object that's been imaged. The precision with which you can localize is much better than the diffraction limited resolution. So starting from 200 or 250 nanometers for resolution, in the same microscope, the localization capability goes as this expression which has the number of detected photons, the starting standard deviation of the point spread function, S, and the pixel size, Q, and the background noise per pixel, B. So you can see that if the background is minimized, then this term will dominate. And if the pixel size is chosen to be appropriately small, what you end up with is a reduction to sigma x equal to S over the square root of n, where n is the number of detected photons. And this can be in the thousands, hundreds to thousands in a typical single molecule microscope. So if you detect 1,000 photons, then you're getting the square root of 1,000, a roughly 30-fold improvement in the localization precision compared to the resolution. So this means that even with a resolution of 240 nanometers, if you detect 1,000 photons, you're in the range of less than 10 nanometers for the localization precision. And in fact, the numbers as good as 1 to 1.5 nanometer localization precision have been published recently. So the question is, even if we can find these objects in space, how do we construct an image out of them? Since imaging one molecule, even with nanometer precision, doesn't give you any real information about the structure. And if you're going to get a map of the structure, you'd like to sample the structure well enough on the length scale of interest. So you're going to need these molecules to be closer together than the diffraction limited resolution in order to get a map of the sample. So then if you make them too close together, you can't localize them, so you're back to the same problem. So the way to get around this is not to look at all the molecules at once. So if you have a conventional microscope image, fluorescence for example, everything blurs together because the molecules are too close together and their point spread functions overlap. And it's like raindrops on a windshield without turning on the windshield wiper. Eventually the density increases to where they coalesce and they're indistinguishable. You can't reconstruct the original pattern. But if you turn the windshield wiper on, then the density can be low enough and each raindrop can be measured with a ruler, and even if the diameter of the raindrop is a centimeter, you can find its location with whatever precision your ruler has. And so the analogy then in microscopy is start with molecules that are not visible and use some kind of control, which will turn out to be optical, but could be chemical or environmental, to turn on a sparse subset of the molecules. And we don't control which molecules. We can't do that. It's quantum mechanics, and that's going to be stochastic. But we can control the number that turn up per frame on the average. And if we make this number small enough that the distance between the molecules is large enough that we can localize them, then we take this set of images and we find the coordinates of those molecules. And then we can turn that off, turn that set off, and turn on another set and localize those and turn them off and repeat until we've localized thousands or hundreds of thousands or perhaps 1 million molecules. And then we plot all the coordinates of the localized molecules as Gaussian spots, usually with a size that's equal to the precision with which we determine the position, and an intensity which is proportional to the number of detected photons. And then we get a map of where all the molecules were. And it's now limited not by the diffraction resolution, but by the precision of localization and also by the density. So this is how we get around the diffraction limit. And two other groups published similar methods essentially simultaneously, PALM by Eric Betzig and STORM by Jiawei Zhuang. And there's also been great developments by Stefan Hell's lab in super resolution. Actually, several years before this localization stuff came out, he had a method called STED, which is more of a point spread function engineering based technique, but you can see it was quite a bit earlier. So what kind of a microscope does it take for this? You might expect it to be something elaborate, but in fact it can be as simple as a standard fluorescence, epifluorescence microscope with a very sensitive camera, that being one of the most expensive pieces of it, and then 2 light sources, which could be lasers, which are convenient that way, that can be controlled with shutters. They get focused, they're parallel here, get focused into the back aperture of the objective. This is not a scanning method, it's a wide field, so you're illuminating a large area at the sample, and then the fluorescence is collected, imaged onto the camera. You might have a telescope in the detection path, but it's basically possible to construct one of these for about 100K from scratch. Much less than a brand new confocal. And the data ends up being a movie, basically, of the molecules as they get activated, and we draw green boxes around them when they're identified by the image analysis method. So then the ones that are identified get localized. You fit their image with a 2-dimensional Gaussian, and that gives you the coordinates and the amplitude. And then you plot those molecules. You could see them building up slowly over time. You build up the structure, and then you can compare with the conventional image and see there's clearly a lot more detail here. And so in a finished example, this is some stress fibers in a fibroblast. So these are actin filaments, which should have a linear form in the form of a stress fiber. If you compare the conventional fluorescence image with the localization-based image, you can see the improvement in the resolution. And so I mentioned that we wanted a molecule which could be initially in a dark state and could be controlled optically. So there are different classes of these, which are generally termed photoactivatable and photoswitchable. And in comparison to a standard fluorophore, okay, so you have excitation light coming in. The standard fluorophore would fluoresce under the excitation wavelength until it bleaches. But our probes don't do that. They're dark, even though we're blasting the sample with the correct excitation wavelength. Nothing comes out until we activate them with a second wavelength. And so that's the stochastic process, and then it causes the molecule to become fluorescent, just like a normal fluorescent molecule. And the molecules then stick around until they bleach. You actually want them to bleach or be deactivated at some point so that the density of molecules as you're lighting up new ones doesn't become too dense and prevent localization. And so that's a photoactivatable, goes dark to bright. Photoswitchable goes from one color to another, switched by a particular wavelength. So if you were imaging the orange fluorescence, you'd see nothing. All of a sudden, you activate the molecule and you see a bright spot show up. But having this standard fluorescent state initially is helpful because you can tell that the probe is there and get a conventional image of it. And there are quite a variety of these probes discussed in literature. This is not a comprehensive list. It's growing daily. But there are a number of different fluorescent protein-based tags, which we like because of the flexibility of the genetic encodability. So you can attach, it's like a GFP, except it's photoactivatable. You've got the ability to control the location and the amount, and the promoter determines the concentration you end up with. You just attach the gene for this protein to your protein of interest and transfect, and the cell does most of the work. Notice that a lot of these are dark to bright, and then a good group of them are one state to another, photoswitchable. And in particular, these orange and red fluorescent ones are good for cell imaging because they avoid a lot of the cellular autofluorescence, and they also give out lots of photons before bleaching. If you don't want to use genetically encoded proteins, you want something smaller, you can use an organic dye. These have actually been around since the 70s, and so in principle, these methods could have been invented back then. We had the capability to image single molecules for a long time, and the photoactivatable probes were around. Why didn't it happen? I'm not sure. But so there are organic dyes, and then Storm uses antibodies conjugated with Alexa or cyanin dyes typically, and there's also a D-Storm method that uses a single fluorescent dye conjugated to the antibody, whereas Storm started off using pairs. And then you can also do a lot with standard fluorophores if you get your conditions right. The Storm type methods often use reducing agents in the buffer, and that allows the molecules to stay in dark states long enough, and it also reduces their photobleaching. So that's something to think about. If you have to add beta-mercaptoethanol to your solution, then that may have some effect on the cell function, whereas these fluorescent proteins can be transfected in with very little effect on the cell function. So what are some of the questions we can address? And the one that's been on my mind recently is how influenza virus infects. This depends on the surface protein hemagglutinin, which catalyzes fusion between the endosomal membrane and the viral membrane, allowing the RNA from the virus to get into your cell. And so the protein HA clusters in order to cooperatively open this fusion pore. So the clustering and the lateral organization of the membrane itself is crucial for the virus's life cycle. And the clustering is also an interesting thing from the point of view of membrane biology. So the concept of a lipid raft was coined in 1997 by Kai Simons as a way to describe heterogeneity in cell membranes. The Singer-Nicholson model proposed sort of a uniform ocean of proteins and lipids, but that model was sort of decided to be not complex enough, and so this Raft model came out, which allows two different types of structures. A raft, which is a ordered lipid phase that's got enrichment in saturated lipids and cholesterol and relatively ordered acyl chains, and a non-raft phase, which is also a fluid and has less order, more unsaturated lipids, and less cholesterol. And the idea is that you'd have protein molecules that preferentially partition into the raft phase and so are therefore clustered in these domains. So that's the model of the membrane, which is based on fluid coexistence. And I say this is a field in controversy because this model has not been accepted as a good explanation in all cases for cell membranes, especially live cell membranes. So we'd really like to be able to get pictures of hemagglutinin in the cell membrane and see what's really going on, but the domain size is typically too small to image with conventional microscopy. And so you can't be sure which of these models is right because they predict different things that you can't detect without the better resolution. This one is a transient confinement model, which invokes interactions between the cytoskeleton and membrane proteins, and those membrane proteins act as diffusion barriers or sort of like fence posts. And then the lipids and proteins that are floating around can't move out of these zones very quickly, so they're transiently confined there. So this is supposed to be membrane-associated actin. And so that has one prediction, right? You'd expect rounded domains in this case because of the boundary between the two phases having an energy penalty, whereas in this case you might not predict round clusters. And then this lipid shell model invokes interactions specifically between proteins and membrane lipids, causing small clusters in the tens of nanometers, which can move around independently. So different predictions, all below the diffraction limit. So if we look in purely lipidic systems, we do see the liquid ordered and liquid disordered phase in coexistence. These are giant unilamellar vesicles composed of a saturated, unsaturated lipid, and cholesterol, and then doped with two fluorescent dyes, parylene and lissamine, rhodamine, DOPE, and imaged by two-photon microscopy. So this is a single membrane bilayer that's in a three-dimensional structure like maybe like a shopping bag filled with water. And you have these phases which spontaneously form. You can see that the domain boundary is round, and that's because the two phases don't like to touch each other. They have to rearrange because of the height mismatch, and that costs a certain amount of energy per unit length. And that's called line tension because it has the units of a force. And from analyzing these shapes, you can see, first of all, that the line tension does play a role. The minimization of the perimeter actually causes these clusters to bulge out of plane in 3 dimensions. And so you can imagine if one particular protein from the virus were enriched in this green phase, which is the ordered phase, that this could help the virus to bud because it would provide essentially free energy to squeeze this perimeter down as the virus is trying to squeeze the membrane into a new sphere to escape. So the fact that there is a line tension in this type of system of about a piconewton is interesting, and the question is whether that happens in a live cell. You also can see some pretty interesting structures that occur just in lipidic systems. So there's no protein here, and yet you've got this enrichment of one lipid in the very curved membrane regions, which could be Golgi or endoplasmic reticulum inside of a cell, and the other probe is in the more straight membrane regions. No protein there. So the interesting thing, though, is when you go to cell membranes, it looks completely different. So the rounded domain shapes go away. There's no circularity anymore. The clusters span a huge range of sizes from the few microns down to below what we can resolve. And we've labeled this in 3 different ways. This is all hemagglutinin, one labeled with regular EGFP, one labeled with an antibody, and one labeled with EYFP. But in any case, it doesn't look in this kind of image like it's easy to measure the domain size distribution. So we're going to move toward super resolution microscopy, but first we're going to do an experiment where we disrupt the membrane cytoskeletal interactions. And we do this by forming what's called a bleb, where you add a chemical to the cell, DMSO, methanol, ethanol. The cell is dying, and its membrane is separating from the cortical actin, which is just below the membrane, causing these blisters to form on the cell and causing a complete reorganization of the membrane. And in these blebs, you do see round phase boundaries, suggesting that two fluids are in coexistence. So this suggests that actin may play a role, because that's the cytoskeleton element that's right underneath the membrane. Actin may play a significant role in organizing the membranes, and this may be the reason that the clusters in cells are not the same as physics and chemistry would predict. So what happens when we look with super resolution? This is a conventional fluorescence image of our viral protein, and here's the super resolution image. So you can see the significant enhancement in the resolution. And if we look at a top-down view now of a sheet of membrane from a cell that's been fixed, the conventional image shows these very amorphous blobs. And when we look in super resolution, you can see that they're not rounded. They're quite jagged in many cases. And certainly they're not minimizing their domain perimeter. So the line tension is not dominating the observed behavior in these cells. Now, that was in a fixed cell. You could look with electron microscopy at a fixed cell, but what happens in a live cell? That's where I think the really interesting questions are. And we were able to push from our initial invention to be the first ones to do live cell localization microscopy. And when we do so, we see a tremendous amount of dynamics. So note these, so this is hemagglutinin again in live HEB2 cells at room temperature. Note these red clusters. I'll play the movie again. These red clusters are everywhere that the HAs were observed throughout the acquisition. This is time in seconds, and this is a 1 micron scale bar. And in this progression, you can see clearly that the molecules are moving around greatly as a function of time. They're moving within the clusters. They're not going everywhere. So why don't they spread around fully into the two-dimensional space? Even though they're moving quite a bit. And how is it they end up with these funny shaped perimeters even though they seem to be diffusing? Well, maybe that was something to do with room temperature, so we went to physiological temperature, and again we see striking amount of dynamics, molecules moving quickly within the clusters, and the clusters themselves mapping out very irregular shapes, and yet some regions don't end up allowing the HAs in. So if we look at time-lapse clustering, time-lapse images of these clusters, we see sort of a full render over 40 seconds shows these linear-looking structures of the HA clusters. If we break it down into 10-second renders, is it just because we had one vesicle that was moving quickly and streaked out this linear thing? Appears not to be, because when we observe over and over again, we see the same kind of clusters persisting over tens of seconds. So something is limiting the motion of the HAs from going laterally, but preferring to have them go along these tracks. And so when we look at the individual trajectories, that's another thing you can do with this kind of a method. You've got the position of the molecule as a function of time. That's kind of the physicist's dream, R and P as a function of X and T. So we can track them over several frames, and we can see what their trajectories look like. And it turns out when we analyze these trajectories that there are actually two different populations, one of which is seeming to go in a free-like diffusion with a mean squared displacement that's proportional to time, and with another population that's moving much more slowly. So this is close to the diffusion coefficient published by Frapp, and then Frapp observed an immobile fraction, whereas we actually see these guys moving, but they're just moving very slowly. And they're confined on a length scale of about 100 to 200 nanometers. So the ability to look at the individual molecules allowed us to discover these two different populations, which would be generally averaged out in an ensemble method. And it turns out that 100 to 200 nanometers coincides with the size of actin corrals as imaged by Moroni and Kasumi in electron microscopy. And so this is cortical actin just below the cell membrane. You see these clathrin-coated pits, and you see these crisscrossing actin fibers, which in principle, if they were anchored to transmembrane proteins, could cause little zones where the proteins are confined. So it's interesting that it's the same size, but we haven't really shown that it's actin yet for HA. One thing that we can do with a single color measurement is we can look at HA, we can disrupt actin, and we can see what result occurs, whether there's any difference. And so we did image HA in untreated cells and in cells that were treated to disrupt their actin cytoskeleton. And we find drastic differences in the cluster morphology. In general, the linearity of the cluster seems to go down. The clusters seem to grow in size on the average. And so there's significant effects of actin disruption on the HA clustering. So the question is, how is actin actually acting? And so to explore this question, we decided to create a two-color FPALM capability. And so we use a dichroic in the detection path to split the light into two channels. We detect the light simultaneously from those two channels, and then take the ratio in the two channels to determine the species. And then we can then label with both PM cherry attached to actin and dendra 2 attached to HA. And this is like a slice through the cell, so you're seeing the edge of the cell here and here. And you see the cortical actin just inside the cell membrane, and you see these clusters of HA in the membrane. So there seems to be a correlation, at least everywhere you see HA clusters, you see cortical actin. There are some actin regions that don't have HA, but there is a correlation. And statistically, we did the pair correlation. In fact, there is a correlation between the two. This is a sheet, sort of a top-down view of the bottom membrane of the cell. And again, you see lots of yellow here where the HA is in green and the actin is in red. So clearly correlation between HA and actin in some regions. And here's the statistical analysis. The pair correlation shows HA and actin are correlated on length scales of a few hundred nanometers. Again, consistent with that actin corral size. And it's true in different regions and different cell types. So this is a kidney cell and this is a fibroblast. So going back to the trajectories, with two colors, we can actually measure the positions of the actins, which are shown here in red, and track the trajectories of the HAs right on top of it. And in this case, we see motions of the HAs along these actin-rich membrane regions. And in regions where there's no actin, you don't see any HA, or generally much less. So if we quantify these steps, we actually find that there is a persistence in the direction of the HA for a few steps. The HAs are much more likely to move ahead in the same direction for a few steps, or to reverse and go at a roughly 180-degree angle from their initial step, whereas it's less likely for them to go at plus or minus 90. And especially in regions of low actin density, they're able to continue a couple of steps in the same direction. But in high actin density regions, they're less likely to keep going, and they're more likely to bump into something and reverse. And the mobility declines as a function of increasing actin density, again suggesting that the HAs are interacting with the actin. They're moving along the actin-rich regions, but they may be bumping into things as they do so. So consistent with the idea of a confinement. Now, if we disrupt the actin and we label both HA and actin, you might expect that the correlation would go away, but in fact, they stay correlated. It's just that the actin regions now have less filamentous character and look more globular, but you still have good correlation between the HA and the actin. So there's something that's drawing the HA into the actin-rich regions. So to sort of summarize what we're learning about the membrane, going back to these different models, so if you had two fluid phases, the classic Raft model, you would explain the motion of the molecules within the clusters. That's fine for a fluid. But the shape of the clusters is inconsistent because we don't really see these rounded domain shapes like you'd expect if it were two fluids in coexistence. Solid phase could explain the jagged shape, but it doesn't explain the motion. So it can't be solid phase because the molecules are clearly mobile. It could be some form of a picket fence model. That does seem to be consistent with our observations. The nanodomain model, which says that the clusters are just a few molecules in size, that doesn't fit. But if you allow the fluid to have some kind of a perturbed perimeter, perhaps because of some proteins anchoring it, perhaps because of wetting by actin filaments just below the membrane, this could explain the linearity of the clusters. It could explain why the HAs have two different mobilities, because some of them are interacting and some of them are not. And the irregular shape could be explained. So this may be sort of this kind of a confinement of the HAs in this kind of a structure might be the explanation for what we see. So I thought I would try this idea out by simulating it, and I just made some very simple rules that the HAs diffuse more slowly when they're near an actin, they diffuse more slowly when they're near another HA, and otherwise they can move around freely in 2 dimensions, and the actins are immobile initially. And then, so you see, you get these linear structures and some globular, and then we disrupt the actin. That's why the red dots are now moving around freely. I allowed them at some point to be disrupted as though we treated with a drug, and you see that the linear clusters go away, and you do get more globular HA clusters that correlate with the actin still. So this simple set of rules explains a lot of what we see. And so the question is only, what could it be that's interacting with the HA? And so to be able to image actin and HA and an adapter protein, we would need to be able to image 3 things. And so we developed also a 3-color version, which uses the 2 channels in the detection path. And it's the same excitation geometry. You just take the fluorescence and split it with a dichroic. And so you get these 2 images, reflected fluorescence and transmitted fluorescence. And so you see the molecules, the same molecules are imaged in both channels. And we use the same ratio as we did in the 2-color case. But now we're just going to have 3 different species. Actually, this movie shows how the data builds up over time. And this is a live cell expressing hemagglutinin with 1 color and transferrin receptor with another color. I'll show some actual 3-color data next, but this is actually 2 species. You can see the 2 peaks building up as a function of time. You can also see a lot of dynamics of the transferrin receptors. They're moving pretty quickly here as a function of time. So when we have 3 species, we have 3 different ranges for alpha that we assign to, this is dendra, so it's like 0.4 to 0.6. This is cherry actins, like 0.6 to 0.75. And PAM Kate, something greater than 0.75, maybe 0.8 and above. And so we can then separate, in general, these species with roughly a 5% error rate. And we can render then 3 different things at once. So we haven't done the adapter proteins yet. We're working on this. But we have done HA, which is a Raft-associated protein, transferrin receptor, which is non-Raft, and actin. And what we see are distinct clusters of HA and transferrin receptor. So those 2 guys don't correlate with each other. But we know that HA correlates with actin. And we also find that transferrin receptor correlates sometimes with actin. So the question is, you've got A correlated with B, A correlated with C, and yet B and C are not correlated. They're anti-correlated. And so you can't just have binding to actin as the rule. It has to be something more specific than that. And so another example of correlation between the transferrin receptor and actin. But our new hypothesis is that there must be some kind of a specific interaction, probably actin adapter proteins, which mediate the interactions between HA and actin. Furthermore, HA and actin have not been observed to interact, or at least they haven't been published to interact directly. And there's not really enough of a cytoplasmic tail for HA to do that anyway. And there's apparently no actin binding domain within HA. So it's got to be something else that's mediating it. But it has to be specific enough to give one kind of cluster for HA that's associated with actin, and another kind of cluster for transferrin receptor that's also associated with actin, but not in the same place. So intriguingly, there was a study in 2008 that looked at purified influenza virus and found a number of actin adapter proteins were present inside of the purified virus. So we're looking basically at this list of adapter proteins to see whether any of those might be the mediator between actin and HA. Okay, so that was a lot about membrane organization, but I think it's important to show that we can really learn something with these methods. It's not just a new tool, it's not just I'm showing a result that we showed that it worked, but we actually were able to learn something biologically. And just to show you that it's not limited to membrane type structures, we're also in collaboration with a group at Brandeis, John Listman's lab, and his postdoc David Santucci brought samples up to our lab of hippocampal neuronal cultures, and we were able to image the dendrites and the spines of each individual neuron. So the green is the wide field image, which is quite blurry, you can see, and the orange is the FPALM, which is much better resolved. In fact, on this putative spine, we see heterogeneity in PSD95, which is normally localized to the synapse, but with wide field microscopy, you wouldn't see any real concentration gradient there, you just see a blob. So we're able to resolve structures within the synapse. And we can even do this in a brain slice. So this was 25 micron thick hippocampal slice, again transfected with PSD95 tagged with Dendro2, and we were able to actually image those molecules at that depth. So that's encouraging that we can do this in tissue as well. And we can do nuclear structure. This is done in collaboration with Mitch Smith at University of Virginia, where we're looking at a protein that tags the histones, which reveals the DNA structure in the nucleus. And these are nice, very well resolved structures of chromatin. So this is a way to check what's going on in the nucleus. Perhaps some of the rearrangements that occur at the onset of cancer could be detected by having this improved resolution. You can also track the mobility of molecules, which could be a reporter on the openness of the DNA structure locally. And that's, again, something that would be hard to do, especially over a large scale. You could do it with a point measurement with FRAP, but to be able to cover a whole nucleus and get mobility as well as an image is hard to do by other methods. And also, I showed lots of two-dimensional examples, but we do have a three-dimensional version of FPALM. And this takes the standard detection path and modifies it to split it in half. So this is just a 50-50 beam splitter. And that first, so the transmitted light just goes straight through to the camera. The other half of it goes to a mirror and ends up on a longer path to the camera. And it's in its image is displaced so that they're not overlapping, they're not interfering with each other, they're just being simultaneously imaged. This longer path length means that what you image in the one image is a slightly different focal plane. So, like your digital camera fixing the focus, taking a picture and then moving it slightly closer to the sample, you're going to have a different focal plane. So you're getting these two focal planes simultaneously, and a molecule that's in the lower focal plane or closer to it will be sharper in the lower image and blurrier in the upper image, and something that's closer to the upper focal plane will be sharper there. So you can fit these two images with the measured point spread function and determine the z coordinate with roughly 70 nanometer localization precision, given the number of photons we detect. This isn't really a fundamental limitation. It's going to go like sigma z over square root of n, and sigma z is your axial depth of field, or your axial extent of your point spread function. So in principle, if you detect more photons, you can do better than this, but that's just a typical value. And we were able then to reconstruct a three-dimensional sample. These are some beads labeled with caged fluorescein. So you can see it really gives a good three-dimensional reconstruction. And all of those stray dots here are actually molecules on the cover slip. So you'll see, if I play this again from the start, you'll see that they're all in one plane there. So you can measure your localization that way too. And it's very important actually to measure, not just calculate. You can use the expression from Thomson, Larson, and Webb to calculate what the localization precision is, but that's the best you're going to do. In reality, what you should do is measure the same molecule in a fixed sample several frames in a row and check. And it's almost always bigger than the calculated, but not much bigger. Okay, so to summarize, we've invented a method called FPALM, which allows a localization-based resolution in the tens of nanometer range. It works with visible light, conventional fluorescent probes or photoactivatable probes. Far-field optics, it's compatible with live cells or other things. We've also done some work on nanostructures. Those are easier, actually, than a live cell, which could be crawling around. And it gives both better resolution and it gives us dynamic information. So we can track the trajectories. It's, as far as time resolution goes, we're at the less than a second, half a second range right now. We're able to generate enough camera frames, so it might be 500 camera frames at 1,000 frames per second to get enough localized coordinates to sample the structure of interest within a half a second. And we were able to learn about the membrane distribution of protein from the flu virus. We found it very non-random, although the range of cluster sizes was quite broad, so not what you'd predict for a simple fluid-fluid coexistence model. In fact, we're basically shooting down 3 models of membrane structure that had been in contention. And what looks like more important is actin interactions with the membrane that seem to inhibit the diffusion of molecules over large distances, or large being hundreds of nanometers. But the question still is, or the new question is, how is it that there's enough specificity to create clusters of many types? Because the cell certainly needs clusters of many types. It can't just put everything into the raft and have enough specificity and function. There need to be specialized types of clusters. And so what's the mechanism for that differentiation? That's what we're working on now. We were able to develop multicolor versions and three-dimensional imaging versions of FPALM, as well as an anisotropy-based method, which I'm happy to talk about if anybody's interested, that can give you some information about the orientation of the label. And if you can attach the label in a well-defined way to the molecule of interest, you can image the orientation of your molecule of interest. We could do this at physiological temperature, which gives us good relevance for a lot of cell biological processes. And I'd point out that this is just one way of doing this. So in principle, any way of creating a sparse distribution, there are other techniques like paint and Fiona, and there's a whole almost overwhelming number of new methods that are based on localization. So it doesn't have to be photo-switchable. It could be that you have a probe, which it does need to be a single-step process, and it should cause the molecule to be activated for long enough that you can get enough photons to localize it. But it could be some other chemical coming in and binding, causing the molecule to switch on. It could be a proton. It could be a timer. I mean, there are proteins that just take time to mature and then they switch on. So lots of ways to create such a method. And I think the good news is that there are many possible applications. I mean, who wouldn't want a factor of 10 better resolution, and also to be able to do this in live cells? So I guess the question is, what is the best thing to look at with these methods? And it doesn't have to be biological. It could be anything you can label with a fluorescent probe. So to thank and acknowledge, there's been really a big team that's helped me to be able to do this work. This is a good part of my lab. Mansa Gudetti just recently finished her postdoc and is now a researcher at a company that's producing these microscopes commercially called Butara. She's heavily involved in the influenza work and especially the live cell work. Travis and Siat were crucial in development of the biplane. Travis also was heavily involved in the HA imaging, and Siat led the 3-color developments. Kristen is doing another collaboration with another virologist on campus, and I didn't get to talk about her work. And Dahan is also taking on and carrying out some of the actin membrane interaction work. I also like to thank these folks for mentoring and useful discussions, and these folks for constructs and probes, and these agencies for funding. And these are also some of my other collaborators. Thanks very much for your attention. I'd be happy to take any questions. Mm-hmm.
The second one has to do with your activation, whether there's any advantage to modulating the activation, looking at different activation wavelengths in order to distinguish.
Yeah, great. So, great question. So the first one, how does the time resolution compare to confocal? To some extent, it depends what resolution do you want. So if you're willing to accept 50 nanometer resolution, which is not the best we can do, you can go 4 times lower in density and still sample the structure well enough. So that means it's 4 times faster. When I report that half second, that's nearest neighbor distance in the, I think it's like 30 or 40 nanometers. So you could go a little faster, and that's not necessarily the best we can do fundamentally, but it's limited partly by frame rate of the camera, partly by how much power you're willing to put on your sample before it gets damaged. So that's certainly an important question. But I guess confocals can go certainly faster than 2 hertz. But in some applications, at least the time resolution of a half second is fast enough to resolve many structures. So if you want to go for the 25 or 20 nanometer resolution, then you need that higher density. So it's going to be a few seconds to do that. Although there was a recent publication by the Lidkey lab that showed that using algorithms from astrophysics for densely packed objects to determine their coordinates was successful at analyzing a 10-fold higher density than what we're used to. So that means using that algorithm, we can go 10 times faster. So maybe we're really able to reach 20 hertz. Absolutely. The second question about activation, yeah. So absolutely an interesting thing to look at. And one of the things I didn't mention as a disadvantage or a complication is that the beam that you use to image the molecules can also activate. And you don't necessarily want this because you'd like to have complete control. And it kind of reduces the amount of control you have. So if you tune your readout wavelength, that can also help reduce the activation from the readout beam. And one of the reasons in the original paper we used 496 nanometer light to excite PAGFP, even though the peak is at 488, or 491, I think. We used 496 because it got us down in readout activation rate, which was quite a problem for PAGFP. For the other probes like Dendra and EOS and Cherry, the readout isn't quite as bad. And so then you could think about tuning your activation wavelength. We've done this, and it tends to get more efficient as you go to the blue, less efficient as you go to the red. And some people are now measuring spectra for activation. So that's absolutely an interesting thing to explore. And in fact, it might be interesting to look at the energy barrier. If you go as a function of energy, you can get into the mechanism. There are some crystallographic studies of mechanism, but the energetics is also interesting. And the rate. It doesn't appear that the rate of activation limits what we're doing. Most of these guys are turning on at about a millisecond.
I did have a question. I'm not sure I understand the sequence of events because you said you are able to reach as high as 500 frames per second or more, yet there is a whole sequence of events that to me seems to be quite difficult to control at the speed. Excitation beam.
Yeah, great question. Yeah. So originally these papers published the canonical form where the canonical sequence or protocol where you say, you turn on the activation beam, you activate some molecules, the readout beam is off. You turn the activation beam off, you turn the readout beam on. You image the ones that were activated. They bleach, you turn the readout beam off. Now you switch back to the activation beam. And it still goes back and forth, back and forth, back and forth. To do this at 500 hertz is more tricky. But you don't need to. That's what we call the synchronous method. But the asynchronous method works great. You just don't have all your molecules doing the same thing. You have a weak amount of activation that's on all the time. You have a strong amount of readout that's on all the time. Some molecules, you get some that have just activated. You see them for a while. You have some that have been on for a while. You have some that are about to bleach. You've got a lot that already bleached. And it's a steady state. You've got new guys coming in, old guys going out, the same number, more or less, per frame, which will gradually go down as you deplete the number of inactive ones. But you gradually ramp up the activation intensity to keep the same number coming in. And it's just open the shutter and take the movie and gradually turn up the activation power. So it's very simple.
Any more questions?
back an image maybe a day or 2 days later, it's going to be the same spot?
Very interesting question. So I guess it would depend on the phase of the material. There are a number of organic dyes that are water soluble, or lipid soluble, hydrophobic or hydrophilic, that are fairly small, but I imagine would stay immobile on at some length scale, tens of nanometers over that period of time. And you could always measure it. Try some and observe the molecules over a few frames. That'll tell you something about the diffusion. You can make the frame time a lot slower, make it a second per frame, turn the laser power way down, and you could still get a picture of what's going on as a function of time of the same molecules. And what was this? Any specifics on the sample?
Just brainstorming about ice right now.
Okay,
I don't know if there's any particular way that it has that.
so we've actually looked at dried crystalline structures where you take the dye and you mix it with some solvent, maybe with some salts or something, and just to make a coating on a surface that has some structure. And it works very well. The molecules are quite immobile on the length scales that we care about. And you can really see pretty cool patterns. We didn't really do anything scientific with this, but just as a way to create some sample to measure, do we get the probe? How does the probe work? What is the photophysics of the probe? But for actually labeling a sample of ice, I think it would work. I think you would just add some caged fluorescein to your water, freeze it, and then you have to keep the sample cold while you're imaging it. So, cryo stage, but it's not that cold. So, interesting question.
All right, well, thank you very much, Sam.